me questions with the recording. That's not a big deal. Okay. All right. Today we're going to begin our A&P2 course by continuing on the nervous system. In A&P1 we covered nervous tissue. That was chapter 10. And then we covered the divisions of the nervous system. That was chapter 11. And now we're going to look at the senses of the nervous system. So this tells you a little bit about the importance of the nervous system. Some of this is just getting through a few little definitions. We're going to be looking at that. There's a lot of terminology in this chapter. It's like a lot of A&P where it's just a whole new language. And your first hurdle in every chapter is to get the words defined. And most of these words deal with structures of on organs. And you'll encounter a lot of that. You know that from A&P1. So once you know where every little part is, then the physiology is not quite so tough if you know the terminology first. So anyway, when we look at our senses, we talked about this a little bit back in the last chapter in A&P1, especially when we were doing the reflex arc. Remember, you had a receptor that picked up a stimulus. It was activated by a stimulus, and then as a result, the nerve impulse was transmitted. Okay, well, these receptors are activated by different kinds of stimuli. And one way to classify them is as to, what kind of stimulus activates it. So we're going to see several today that are classified as chemoreceptors, and that means they come in contact with a specific chemical. And if there's enough of that chemical, it will cause an action potential to occur. Pain receptors, in your book, they may give you an additional name for these. I won't put this one on your test necessarily, but a nociceptor, that's a pain receptor. And they're actually sensitive to a lot of different things. We'll see that when we look at pain in a little while. Some pain receptors are activated by the physical tissue damage, some by chemicals that are released as a result of tissue damage, some because of fluid pressure that's a result of the tissue damage. There's a lot of different things, but we just classify it all as pain receptors. And then a thermoreceptor, that's kind of obvious. It's sensitive to changes in body temperature. So some of those are sensitive to elevated body temperature. So those would be your hot receptors. And then your cold receptors would be sensitive to decreases in body temperature. And then I love these. Mechanoreceptors are sensitive to a change in the shape of the receptor cell, basically. So a mechanical force, not a chemical, but a mechanical force. We're going to see some of these. Sometimes you have receptors that get stretched, and we'll see a couple of these today. They're called proprioceptors or stretch receptors, and if the cell itself gets stretched enough, that triggers an action potential to occur. All right, and then some of them are sensitive to pressures, like you've got some in your bloodstream or in the wall of some of your blood vessels that are sensitive to changes in blood pressure. And we will see those specifically when we get to the cardiovascular system. We'll see how they affect that change in blood pressure. In turn affects these receptors, which in turn affects your heart rate, and which counteracts those changes in blood pressure. And then some other mechanoreceptors we'll see this morning, that like there are some that the cells have cilia on them. And if those cilia get bent for some reason or another, that bending, which is a change in shape or a change in position, is enough to trigger an action potential. Right? So just some physical type movements trigger these. And then photoreceptors, of course, these are only found in the eye, and they're sensitive to light waves. Okay, now when we talk about these receptors, there's a couple of ways they can get activated once they get in contact with the stimulus. Sometimes the receptor is the actual dendrite of a neuron, of a sensory neuron. Okay, and so it's just stimulated directly. But other times the neuron, the dendrite ending of the neuron is connected to another cell. Oops, let me go back. For example, let's say here's the end of the dendrite end of a little neuron, and it is embedded in another cell. So that green would be, say, an epithelial cell. And let's say stimulus affects that cell, and because it's a cell membrane, it's polarized just like neuron cell membranes are polarized. So the stimulus could trigger a change in the permeability, which causes ion movements. But if enough of that occurs, then that's going to trigger an action potential on the neuron. So that would be more of an indirect stimulus. And so sometimes you have that. We'll see both examples of these. In either case, the stimulus must be of the threshold amount. Somebody define threshold for me. Yes. All right. She said it's the minimum amount of stimulus required for, in this case, an action potential. When we did threshold stimulus for muscle contraction, it was the minimum stimulus required to get the muscle to contract. Okay. So if it's a subthreshold stimulus, that means an action potential will not occur. But if it's threshold or superthreshold, it will occur. Okay. All right, and then a few little terms. You know these words. Sensation is, when all of these receptors get activated and an action potential is sent, it's going to your central nervous system. And the bulk of that passes through this middle part of your brain, through that diencephalon area and the thalamus. Remember, that's a relay for incoming sensory impulses. And that gives you that first feeling that something is going on. Okay, so that feeling is called sensation. And then the way you interpret that feeling, like, oh, I feel something is touching my skin of my thumb rather than my elbow. You're knowing, let me back up. Perception is when it's like if something like a gnat lands on your skin, you feel that as a pressure stimulus on your skin. Okay. When you see, that's a visual perception and not an auditory perception. So it's a feeling, but you can direct it a little bit better. And then projection tells you where it's coming from. Like when you hear, it actually feels like you're hearing with your ears. I know that sounds silly, but you're projecting where the stimulus is. If your muscle hurts because you've exercised a lot, you're projecting it at that particular muscle group, okay, and not another one. Okay. And then sometimes when you're exposed to the same stimulus over and over and over and over, that results in those receptors having an even higher threshold. So that same degree of stimulus is not going to activate it anymore. A classic example of that is, let's say you go, how many of you, when you go to your grandparents' home, it just smells like their home? It has a unique fragrance to it. Or when you get in your car, that new car smell, you first get in, but then once you're there for a while, you don't notice it anymore. It's not because those particles are not in the atmosphere. It's simply the threshold has risen. So it would take even more of that. Okay. And so that's called adaptation. And a lot of our senses are adaptive. Okay. Now, there's, in your book, this chapter describes the two major groups of receptors. And, we can call the chapter your senses, but basically you've got two kinds of senses. You have your general or somatic senses, and then you have your special senses. And your general senses are individual little receptors that are just sort of scattered all over the body, like your hot receptors, your cold receptors, your touch receptors in your skin. They're not isolated into one organ. They're in lots of different places. And we're going to look at a lot of those today. But then your special senses, like when you think of your senses, that's what you're thinking of, the smell and taste and hearing and vision. Those are your special senses. And we call them special because the receptors are parts of specific organs. They're not widespread all over. Okay. And so the bulk of your chapter is dealing with your special senses. But we're going to start with the general senses. And we can, just like this is A and P, so there's a lot of ways to describe and term everything. And one way we can describe these is based on where they're located. If they're at your skin, they're referred to as exteroceptive, like all your touch receptors, your hot, your cold receptors, your itch receptors, all of those are at the skin. So those they would be considered exteroceptive senses. Proprioceptive, now we've seen that word this morning. Proprioceptor was a stretch receptor. All right, those stretch receptors are found in your muscles and in your tendons. Okay, but so those senses sometimes are classified as proprioceptive senses, and then visceroceptive senses are in all your other internal organs. Okay, viscera, remember, means internal organs. Okay, another way we can group them is based on the kind of stimulus. So we've seen that a little bit. So now you understand what a chemoreceptor is and a pain receptor. You understand a little bit about it. And now we're going to go into that in a little more depth. Okay, so we're going to start with some of the mechanoreceptors that are distributed in lots of places that allow us to experience touch or pressure in places. So our touch and pressure senses. Some of these are just some free nerve endings, like for itch. When your skin itches, a lot of different things can activate that. But they're free nerve endings. But these next two we're going to see in lab when we go into lab today, a Meissner's corpuscle and a Pacinian corpuscle. Okay, so what I'd like for you to do is stay on this slide, and I want to move to a picture. Y'all remember this picture from the skin chapter? And where here's your epidermis, and then this is the dermis, and then there's the subcutaneous layer. Well, within the dermis, there are a lot of somatic receptors. And a lot of that yellow that you see, a lot of these are neurons. And let's see if I carry this one up. There's the little dendrite end of a neuron. And then there's a bunch of connective tissue cells that are wrapped around it like that. This is what you're seeing right here, right there. And there's another one right there. And that is called a Meissner's corpuscle. So what that is a neuron ending, and then it has connective tissue cells surrounding it in kind of a tornado-like fashion. Looks like a little tiny tornado in there. I want you to notice that these are located up high in the dermis. Like, I know that's not one, but if there was one, it would be located in there. All right, in this area that's high in the dermis, you see where the dermis projects up towards the epidermis? That little upward projection is called a dermal papilla. Papilla just means upward projection, okay? And Meissner's corpuscles, most of them are located in the dermis of the skin within dermal papilla. So in other words, very high in the dermis, not the epidermis. They're high in the dermis. They're about as high as you can get in the dermis. Okay, and they are sensitive to very tiny change in position. So in other words, if they get squished a little bit, if these connective tissue cells get pressed upon and they change shape a little bit, that might be enough, if it's the threshold amount, to trigger an action potential. So this would be one of those indirect stimuli. All right, but it's sensitive to, this is in your notes on the slide before this, to light, touch, and also to texture. Okay, so and they're in your skin. Areas in your skin that are super sensitive to light touch and to texture, like your fingertips, are super sensitive. They're going to have a lot higher concentration of Meissner's corpuscles than areas of your skin that are not as sensitive, like your elbow. Wouldn't be as sensitive to touch maybe as your fingertips or your shoulder. Okay, and if you rubbed your shoulder along something, you couldn't feel its texture as well as your fingertips, could you? okay, because you don't have as many Meissner's corpuscles per square inch in your shoulder as you do in your fingertips. Okay, so the more sensitive an area is of the skin to touch and texture, the higher the concentration of Meissner's corpuscles there. Okay, here's the real thing. We'll see it under the microscope today. All right, then deeper in the skin, and on this slide it doesn't show it very deep, and it also shows it very small, but I'm going to draw another one. I'm going to draw it deep. Sometimes it's down in the subcutaneous layer, but it's a neuron ending, and it has connective tissue cells around it, but this time, instead of being in a swirl, they're more concentric layers of cells, and it actually looks like a sliced onion under the microscope. It's very easy to recognize. It's usually a whole lot bigger than the Meissner's corpuscle. It's called a Pacinian corpuscle, and its location in the skin is down low, deeper in the dermis, maybe even down in the subcutaneous layer, and it is activated again when it's squished, but it would take deep pressure to do that, or a minute vibrations. I don't mean vibrations like this, just minute, almost undetectable vibrations could activate a Pacinian corpuscle. So when you have like a blood pressure cuff on your arm and you feel that pressure, it's activating Pacinian corpuscles. Okay. This sounds silly, but you ever noticed when food is in your mouth, you actually can feel the food touching the wall of the inside of your mouth, the wall of your cheeks. But once you swallow it and it gets down in your stomach, you cannot feel it touching the wall of your stomach, can you? It's still there, but you don't have Meissner's corpuscles in the walls of your stomach, so it's not going to detect that light touch. You do have a lot of them on the inside of your cheeks, and so you feel it there. But if you have a lot of food in your stomach, or your stomach starts grumbling and vibrating some, you feel that because you have Pacinian corpuscles in the walls of your stomach. So typically, your Meissner's corpuscles are only going to be in the skin. Pacinian, you can have Pacinian corpuscles in the skin, but also they could be exteroceptive in the skin, but also visceroceptive. Some of your organs have them. Okay. Then our temperature senses, those are just some free nerve endings, and I mentioned earlier, some of them are activated by temperatures above 98.6 degrees Fahrenheit. Those would be hot receptors, and the cold receptors would be below. And then pain receptors, they're also free nerve endings, and they're pretty much everywhere except within the brain. Now, you can have a headache, but that usually is because of blood vessels around the brain, and you do have pain receptors there. But the nervous tissue itself doesn't have any. But everywhere else, you've got pain receptors, and they're sensitive to all kinds of things. You ever noticed if you touch your hand on a stove, it doesn't really feel hot, it just hurts. Because extremes of heat or extremes of cold activate pain receptors. Okay. A lot of times tissue damage causes certain chemicals to be released or not, and so the presence or absence of those chemicals could activate a pain receptor. And remember I talked about adaptation, that repeated exposure to a stimulus. Sometimes it causes thresholds to rise. With pain receptors, it's the opposite. When pain receptors get activated, thresholds might even lower. Some of these chemicals cause them to lower. So you notice that if you have, say, a bruised area in your skin and you touch it, it hurts. But if it's not bruised and you touch it, it just feels like touch, doesn't it? And it's just that pressure stimulus. Sometimes pressures can activate these pain receptors. Okay. So pain receptors have very little adaptation compared to all the others. Referred pain, you've all heard of this. What are some of the classic warning signs of an impending heart attack? Chest pain is one. In your arm, you ever heard of the issues with most time it's left arm, but it could be the right. The jaw, you ever heard of jaw pain associated with heart disease? All right, that's all what we call referred pain. And Sometimes your brain gets its signals confused. Impulses, like here, imagine that this is the skin of the left arm. Impulses coming in from there end up along similar pathways as impulses coming from the heart. And sometimes the brain misinterprets that. And that's this referred pain. There's nothing wrong with the arm, but that's an indication that there might be something wrong with the heart. And so in the medical community, they learn to recognize these areas of referred pain. You can see this here. You don't have to know these areas for the test, but you might be familiar with some of this. Sometimes when people have a gallbladder attack, it hurts in their back, like under a shoulder blade sometimes. But anyway, those are areas of referred pain. Okay, now pain pathways. It's interesting when you experience pain. There's actually two kinds. There's the acute pain, and then there's chronic pain. So acute pain would be like if you got cut or if you fell, you broke a bone. That immediate sharp pain, that the reason it's acute, one reason, is that the fibers are myelinated. So these are your fast pain fibers. They're myelinated, so that means they conduct impulses more quickly. And as soon as they quit being activated, or as soon as you don't have that stimulus anymore, it's going to go away. But as far as your test is concerned, I want you to know that your fast pain, myelinated, and it's acute, okay? Myelinated, and it's that sharp pain, that acute pain, that tends to be localized wherever the impulse is coming from, where the stimulus is. Whereas your slow pain, that's more chronic. Do I have the word chronic? Yes. Like when you have the flu, you can't pinpoint one spot where it hurts, can you? It's just kind of all over. Some diseases lend themselves to chronic pain. People who struggle with cancer have a lot of chronic pain. It's not just in one spot. It's slow pain fibers. You don't have to know A, delta, and C. Just know slow and fast. And basically, the chronic is the long, dull, aching pain that's very difficult to localize, whereas your acute pain, you can localize that. Okay. And anyway. So I'm not going to test you on details on this, but just know that there's the chronic pain and there's acute pain. Okay. And what the difference is as far as how it feels. Acute pain is that sharp, you can pinpoint it, but it can go away pretty quickly. And then the chronic pain is that dull, aching pain. All right. Okay, then if we look at these proprioceptors or stretch receptors, there's a couple of these, and they work oppositely. So that your brain is aware of the contraction or the stretching of muscles. All right? And so one is called a muscle spindle, and the other is called a Golgi tendon organ. And let's just look at this. On this one, on the left, you're seeing the muscle spindle, which is what all of this is. And do you see how it's within a skeletal muscle? So you see all these other skeletal muscle fibers? But then you see a special group of them, like this one, this one, this one, and this one, that are wrapped by some special connective tissue, and they also have neuron endings wrapped around them. All right, now just imagine if this muscle started stretching. It gets long and thinner, doesn't it? Do you see how the muscle spindle stretches also? Okay. What's going to happen if there's no actual control over muscle stretching? If a muscle just stretched and stretched and stretched and stretched, eventually what's going to happen to it? It's going to break apart, wouldn't it? It's going to tear. All right, and so muscle spindles help prevent that because they detect too much stretch. And if they get stretched to the threshold amount, then they're going to cause an impulse to be sent on this neuron to the brain, and then the brain's going to send an impulse back to the muscle so it will quit stretching. Okay? Because you don't want the muscle to rip apart. That's the muscle spindle prevents too much what? Stretch. Spindle and stretch both start with an S. It doesn't cause the muscles to stretch. It prevents too much stretch. This is the Golgi tendon organ, and it's located within a tendon, like it says, whereas the muscle spindle is in the muscle. But remember, tendons are bands of connective tissue connecting a muscle to a bone, right? Let's say here's the bone that the tendon's connected to. All right, and so if this is for my online students, so they can't see me pointing. That's why I'm writing some of this. But anyway, so there's a bone that this tendon is connected to, and what happens when a muscle contracts and gets shorter, like those isotonic contractions? It gets shorter. What do you see how it's pulling on the tendon? Remember that? It pulls on the tendon, and if it kept contracting, it's going to rip that tendon off the bone, won't it? All right, so a Golgi tendon organ is there to help prevent that. So as a muscle contracts and that tendon lengthens, that's going to activate the fibers in the Golgi tendon organ, which is going to send an impulse to the central nervous system to tell it the muscle's contracting too much, and now the nervous system's going to counteract that by slowing down the contraction. All right? So the Golgi tendon organ prevents too much contraction of the muscle. Okay. All right, so that's all of our little somatic senses. Now we're going to focus on the rest of this chapter, which is your special senses. And the first of those is your sense of smell, which is olfaction. If you hear about olfactory anything, that's smell. Okay? And so you know that this is located somewhere around your nose, don't you? Okay. These receptors are chemoreceptors. In order for you to smell a fragrance, there's a chemical that is actually going to bind to the receptor cell. And the presence of that chemical on the receptor cell is going to activate a nerve impulse. These are gaseous particles in the air that we breathe, and they're dissolved, but then they bind to particular receptor cells. The olfactory organs are actually located all along the upper part of the nasal cavity, not your nostrils. Your nostrils lead to the nasal cavity. Okay? And do you remember in lab in A&P1 when we were studying the skull? Do you remember how you looked in the nasal area and there were 3 shelves? Those are called nasal conchae. Okay, the upper nasal conchae is actually part of the ethmoid bone. The other 2 are part of the maxilla bone. But anyway, along the upper nasal concha is where some of the olfactory organs are, and it kind of continues down the nasal septum just a little bit. So it's all up there in the upper part of the nasal cavity. All right, stay on this part. I want you to look at that. Let's look at a picture, and we'll see some of this. Okay, here's the underside of the brain. And one of the last things we did in lab in A&P1 is talk about a few of the structures on the brain. And one of those was the olfactory nerve, which is your first cranial nerve. And at the end of the olfactory nerve, it enlarges a little bit. That enlargement is called an olfactory bulb. And this olfactory bulb, that right there that I'm circling, sits in the nasal cavity, sits right on top of the nasal cavity right there. So here you see it. Okay. So do you remember when you studied the skull in A&P1, if you took the top off of it, do you remember at the top of the ethmoid bone there was this little ridge that went up called a crista galli, and then on either side was a little perforated plate called cribriform plates? Well, the 2 olfactory bulbs sit on those. And so there you can see it right there, sitting on the cribriform plate. And here's a magnified view of it. So this is the olfactory bulb. And do you see how it has neurons that extend through the holes of the cribriform plate? See, here's the cribriform plate. Okay? And these neurons extend through that, and they hang right there in the top of the nasal cavity. They're the only neurons, I know I've told y'all this in A&P1, but they're the only neurons of your body that are exposed to the outside world. Do you see how they end with little cilia? These are some bipolar neurons. Some of the bipolar neurons, they're mostly in some of your sensory organs. Like here, they're bipolar neurons. And here's the dendrite end of it. And the end of the cell membranes there have molecules on them that gaseous particles are attracted to. And so if enough bind, then that's going to trigger an action potential on this bipolar neuron, goes up through the cribriform plate into the olfactory bulb, which then goes along the olfactory nerve to the smell centers of your brain. So on this slide right here, you can see that pathway. So from the olfactory bulb into the olfactory nerves, and once the nerve gets to the brain, it's called an olfactory tract. It goes through your limbic system. Remember, that's in the diencephalon, and to the olfactory area of your brain, which is not an area we talked about in AMP1, but it's at the bottom of the frontal lobe of the brain, which is there close to the nose. I think this is super interesting. We tend to lose about 1% of these receptors every year. Why do you think we lose them? Because they're exposed. They're neurons. If they are damaged, they don't regenerate, right? They're neurons of this. But they do, even though they're neurons, they have a little bit more regenerative ability than most neurons do. So without that feature, we would lose a lot more. So you basically lose a lot of your sense of smell every year. This last statement, the sense of smell drops by 50% within a second. That means it's highly adaptive. Your sense of smell is very highly adaptive. But you tend to lose your sense of smell as you get older and older and older. Okay, here it is again. I kind of got crazy on some of these. But you can see the olfactory organs right there at the top of the nasal cavity. All right, there's the real thing under the microscope. It's very difficult to tell. It's very difficult for me to tell. This just looks like ciliated epithelium, doesn't it? And I can't tell the neuron from the surrounding epithelial cells. But you do see some of the cilia right there. All right, so that's your sense of smell. Now we're going to look at your sense of taste. Most people know that the organs of taste are called taste buds. However, a lot of people mistakenly think that the taste buds are those little bumps on your tongue. How many of you thought that? You're honest, aren't you? But those bumps on your tongue are not taste buds. Taste buds are microscopic. Those bumps on your tongue are called your tongue's papillae. We've already seen that word today, haven't we? Papilla was that upward projection of the dermis. All right, here's that bump on the tongue. It's an upward projection. So the word papilla just means upward projection. So let's say if there's a tongue's papilla, the taste buds are little microscopic structures located on the sides of some of your papilla, or if there's another papilla, they may be located down at the base of the papilla. Okay? If we let's look at a picture and we'll see all these parts. I will come back to all of this. Hang on. Okay, so here you see kind of a strange looking papilla, but do you see the tiny little dots on the sides? Those are the taste buds. And then here, if you see a taste bud magnified, all right, you can see that there's epithelial cells, just normal, looks like columnar epithelial cells. And then you have specialized epithelial cells that are the actual taste cells. And they have little microvilli that look more like cilia, actually. Your book calls them microvilli, but we refer to them as taste hairs. It's not true hair. It's not keratinized cells. But we refer to them as taste hairs. But on the ends of those are chemicals, like little proteins, that specific particles, specific other chemicals will bind to. And whatever chemical binds, for this particular one, it may give a sweet sensation or a sour or a salty or a bitter. All right, so you have different kinds of taste cells that are specific for certain types of substances dissolved in the foods that you eat. Okay. if enough binds, then it's going to activate this cell, which in turn is connected to a neuron, and so an impulse will be sent on a neuron to the taste centers of the brain. Okay. So would this be direct or indirectly stimulated? Indirect, because the actual stimulus is the chemical, and it's binding to the cell that the neuron is connected to. Okay. Okay. So let's back up and get some of this. Okay. So notice that your food molecules or ions must be dissolved in water before they can bind. Okay. That's one of the many reasons we have saliva. You ever notice when your mouth is really dry, things don't taste as good? You don't notice the taste of food as much, but if you get it wet, you notice it. So, we have 5 primary tastes. We all know about sweet, sour, bitter, and salty, but fairly recently they've identified umami, which is it's more of a meaty, pungent taste. Not necessarily meaty. I don't know how to describe it. A lot of the Thai foods have that. MSG, ever heard of monosodium glutamate? It activates that. All right. Okay. Taste buds, unlike your olfactory cells for smell, taste buds can regenerate much more easily than your smell receptors. And it's because the cell that is coming in direct contact with the stimulus is an epithelial cell, not a neuron. And epithelia has the ability to undergo cell division, right? So as long as the damage doesn't go all the way down to where the neuron is and damage it, then that taste bud can regenerate. You all probably realize that your sense of taste is very strongly related to your sense of smell. If you have a cold and you got so much mucus up there that nothing can bind to the olfactory receptors, food just doesn't taste as good, does it? I think I'm just throwing this out there. I have no idea, but it makes sense to me that one reason as we get older we tend to enjoy foods that don't smell so good, like broccoli and turnip greens. I mean, most little children won't eat turnip greens, and I would think it's one reason is because their sense of smell is so keen. But as you get older, it's not quite. It doesn't bother you quite as much. Anyway, that's not a big deal. All right. Very seldom do I go through something in our lectures that is not found in your book. But this is one topic that is not in your book, is the different kinds of taste buds. The different types of papillae. I just think it's really interesting, and I learned it way back when, and so I wanted to add it to your notes. But anyway, you basically have 4 kinds of papillae. The most abundant one is called a filiform papilla, and if I was going to draw it real magnified, I would do it like this. See how it's jagged on the surface? There are no taste buds associated with that one. But this is the most numerous. So the whole reason for your papilla is not necessarily for taste buds. Okay. The biggest reason for the papillae is that this one anyway, is that it provides friction. So it literally can grab onto your food better, so your food's not sliding around in your mouth. Okay, and that's because it's jagged on the surface. See that? Some mammals have huge filiform papillae. It's like a bunch of them fused together, and they're super long, and they use them in grooming. Can you think of a mammal like that? Cats, especially cats. Yeah, and they have those long, what we call hairs on their tongues. Right, they're really large filiform papillae. The other 3 kinds do have taste buds associated with them. The fungiform, it's called a fungiform because it looks like a little button mushroom. Do y'all know what I mean when I say a button? An immature mushroom, when it's first developing, looks like this. It's called a button mushroom, before the cap opens up. Anyway, a fungiform has that look to it, and that's why it's got the word fungiform. A mushroom is a fungus. Okay. But anyway, taste buds are along the sides there. You tend to have a lot of these isolated at the tip and the edges. Right. Vibrate papillae, that was that weird one we saw on that picture. I can't draw that one. You can see it real well here. I've circled it in green at the top picture. So it's kind of like a fungiform with a ring, another one around it. But you see there's a lot of taste buds associated with it. You've got a row of these at the very back of your tongue. So you're not as aware of them, but today when you look in a mirror, stick your tongue out as far as you can, you'll see some bumps on the back of it. And that's what these are. Okay, the vibrate papillae. And then the foliate papillae, you don't see on this picture. They don't really look like any of the other papillae because they're not upward projections. I'll come back to that. You do see it here. See these folds on the sides like this? These folds right there. I'm not drawing it right. This way. Those. Right there. These folds. Those are the foliate papillae. Foliate. It's like leaves. Foliage, that's where the word comes from. Leaves, foliage, foliate papillae. And there are taste buds in the creases along the sides of that. All right, now this picture is not in your book. And it's been somewhat discounted, but it's still, it's fairly accurate. And I do want you to know this. The taste receptors in the taste buds along the front at the tip of the tongue tend to have a high concentration of the little receptor molecules for disaccharides. And so if the disaccharides bind there, that's going to elicit the sweet taste, okay? And they tend to be towards the front. Sour things like acids, like in lemon juice, is kind of sour, dill pickle juice. That's going to activate taste buds at the side because the receptors for acids are in those taste buds. Some of the foliate papillae have those. Okay. Salty is all along the rim, so the tip and the sides. And then bitter, there must be a lot of bitter receptors within the vibrate papillae at the back. Okay. So do know those areas. Don't worry about you, Mommy. It's just kind of all over. But okay. So those were the two kind of simple ones that don't take much time, olfaction and taste. All right. Now, the rest of today, we're going to spend the rest of our time on the ear. The ear's a big deal. There's a ton of terminology with the ear. There's a lot of different parts to the ear. But it's surprising when I tell you this, the reason you hear something is because cilia bend in the inner part of your ear. Okay? And if the cilia bend enough, that's going to trigger an action potential. Okay, and so before we look at the details of the parts, I want to give you an overview of how the ear works. What happens is there's a sound wave that comes through. This is the part of the ear called the outer ear. Comes down the ear canal, and then it hits this famous structure. We call it the eardrum. In this chapter, we call it the tympanic membrane, but it hits that. And so that sound wave then is channeled into a physical movement, a vibration of your eardrum. And on the other side of the eardrum are three tiny little bones. It's a little bridge of bones. And so as the eardrum vibrates, that bridge of bones moves back and forth and back and forth. And then the third of these bones right here is attached to the inner part of your ear. On this picture shows up as blue. That's a bony structure that is filled with fluid. And that last little stapes, the tiny little bone, as it's vibrating, it pushes against a little membranous opening of this inner ear, which sets up fluid movement. And if that basically the fluid starts moving inside here, the hearing receptors are in this part, and if that fluid moves enough, it's going to ultimately cause, watch, cilia to bend. And those cilia are of cells that are connected to neurons. And that will trigger an action potential to go along the vestibulocochlear nerve to the auditory center of the brain. Isn't that weird that hearing is just because of the bending of cilia? Yeah, he said once you get older, you lose the cilia? Is that why a lot of the hearing diminishes as you get older? That could be part of it. There's a lot of different reasons for hearing loss, but it could be damage to those cells over the years. Yes, definitely. Especially if you've had loud sound exposure, because that makes them, it could damage those cells. Okay, so we're going to talk about these three parts of the ear. You've got the outer ear, the middle ear, that's where that bridge of bones is. It's over through an air-filled space. And then this blue part on my picture is the inner ear. So we're going to take all that one at a time as we look at hearing. Okay, the outer ear, the different parts of the outer ear, there's three structures associated with it. The auricle, and they all have different names. I apologize. It's also called the pinna. That's the part of the ear you see on the side of your head. Okay, so that collects the sound waves. And then the ear canal is also called the external acoustic meatus or external auditory meatus. It's changed names through the years, but any of those will work. External auditory meatus. Y'all remember that when we did the skull? Okay, it's in the temporal bone. That's your ear canal, so the sound waves go through that, and then it hits the tympanic membrane, which is the other name for your eardrum. You have some wax glands that line your ear canal. They're called ceruminous glands, and of course that's to trap foreign things that might be in the air that would interfere with that sound wave conduction. Okay, so that's the outer ear. We're going to look at all this more closely on a picture in a minute. The middle ear is that cavity that has the bones going across it. So it's called the tympanic cavity. And then the bones are the auditory ossicles. Os means bone, auditory ossicles. These are the smallest bones of your body. And there's three of them. They are the malleus, the incus, and the stapes. You pronounce that stapes, not stapes. Stapes. And you need to know the order. The malleus comes first. That's the one connected to the tympanic membrane. The incus is in the middle, and the stapes connects to the inner ear. Okay, so MIS, malleus, incus, stapes. Okay. All right, and then you've all maybe heard of your Eustachian tube or auditory tube. It is a channel that connects the tympanic cavity to your nasopharynx. Pharynx is a fancy name for your throat. The part of it that you see when you open your mouth is the oropharynx, but just above that, above the back of the roof of your mouth, is the nasopharynx before it gets into the nasal cavity. And the Eustachian tube opens into that nasopharynx. So we'll see that. And then the inner ear is a real complicated little structure, and we'll look at its design in a little while. Okay. So here's a repeat that gives you just more information, but it's a repeat of the parts of the external ear. They're in red. The auricle, which is this part, and then the external acoustic meatus is the ear canal, and then the tympanic membrane you can see right there. Okay, tympanic membrane. Okay. The auricle, you know, some animals have really big auricles, like a rabbit. Have y'all ever really watched a rabbit? They can move them independently, and they're moving to collect sound waves. It's just like when you cup your ears, and you basically are making for a bigger auricle, and you can hear better when you do that, can't you? Because it's collecting these sound waves and then channeling them to a physical movement of the tympanic membrane. And then the middle ear, you've got the middle ear cavity, the auditory ossicles, malleus, incus, stapes. You may have heard of them referred to as the hammer, the anvil, and the stirrup. Okay. And then the oval window, that's the little membrane that the stapes is attached to of the inner ear. Okay. So it's kind of part of the stapes / inner ear. Now, the Eustachian tube is what you see on this picture right there. Okay? And it is an air-filled tube. and it connects your tympanic cavity to your nasopharynx. And so air can move up and air can move down. And it does that constantly to keep the air pressure in the middle ear cavity the same as the air pressure in the ear canal, or atmospheric pressure. Okay? And so if you think about it, if the air pressure is equal on both sides of the tympanic membrane, that means the tympanic membrane is not being pushed more in one direction than the other, and so it's as flexible as possible. But if the air pressure was, say, higher out here, do you see how that would push on this and it wouldn't be as flexible? Or vice versa, if it was higher inside, it would be pushing it out a little bit and it still wouldn't be as flexible, wouldn't move as much in response to a sound wave. All right, and so this helps your hearing because it keeps that tympanic membrane as flexible as possible, because it's going to be in an environment where the air pressure is equal on both sides. Sometimes you hear your ears pop, don't you? What you're hearing, what you're experiencing, is a quick rush of air up or down the eustachian tube, not the normal movement of it, but just a quick rush. And so you might experience that. Let's say if you're in an airplane, they have those pressurized cabins. All right, and when you're going up high, you experience a pretty drastic change in the air pressure, so your ears pop. What can you do to minimize that? Have you ever chew gum? You ever notice when you chew gum, you are opening up your jaw, which is basically allowing air to move more easily up into the nasopharynx. And so that does help with it a little bit. How many of you have young children at home? When you take them to the doctor, not their normal checkups, but just the times you just have to go to the doctor because they're sick, some children really struggle with otitis media. Otic anything means ear. This is middle ear. Otitis media is a middle ear infection. So that means in the tympanic cavity. And you see that quite often with children, but rarely with adults. You ever notice that? That kids grow out of having ear infections? All right, that all has to do with their eustachian tube. When you are a little tiny person, they put those right there. He was asking about the tube. We're going to talk about that in just a minute. He was asking about the tubes that they put in a child's ear who has chronic middle ear infections. Anyway, in a child, the eustachian tube is more horizontal. Think about a child's face shape. The lower half of the face is not as elongated as it will be when he gets older. All right, and so that part of the face includes that eustachian tube, so it's more horizontal. So you can imagine if it's horizontal, then bacteria will and other pathogens will more easily get into the middle ear cavity, setting up infection, because they don't have to crawl up against gravity. But also, whenever an infection occurs, inflammation can occur, and during inflammation, excess fluid is produced. And here, fluid's going to be produced in an area that should be just air. And so if that fluid builds up, it causes the pain receptors. But do you see how that fluid can push against the tympanic membrane from the backside? And again, it's going to limit its movement. So over time, it could cause some hearing loss. All right, and so what they do, first of all, as that fluid builds up, in a child, it can't easily go anywhere. In an adult, it could drain down this way, couldn't it? But in a child, it's more horizontal. It's harder to get rid of the fluid to alleviate the pressure. So in the meantime, what they do at the clinic is put tubes in that child's ear. And it's tiny little plastic tubes they just stick in the eardrum. And it just provides a route for fluid drainage, and which prevents fluid pressure on that child, hopefully to prevent some pain, but also hearing loss down the road. Eventually, those little tubes pop out. No, they just pop out on their own. It doesn't really have anything to do with this, but they'll just come out, and the tympanic membrane can heal itself. If you ever have a ruptured eardrum, they don't do anything about it. It repairs itself usually. Um, but then as that child gets older, they literally can grow out of ear infections, and it's just because that eustachian tube becomes more elongated, more angled down. That's pretty interesting, isn't it? Okay. All right, so we've talked about a sound wave hitting that eardrum. So just imagine as that eardrum is vibrating, it's going to push on that bridge of bones that in turn is going to push on a little membranous structure called the oval window, which is going to set up some fluid movement. All right, and so I want you to notice on this, I'm going to back up on this picture. Let me erase this. Okay. I want you to notice the diameter of the eardrum compared to the diameter of that little oval window, which is bigger? The eardrum. So can you imagine that a little bit of movement of this eardrum, a tiny movement, which is what a sound wave causes, minuscule movements of the eardrum, because that's channeled into a smaller diameter area, it's going to cause a bigger movement of the tympanic, of the oval window, which then is enough movement to set up the fluid moving in the inner ear. Okay? It's because of the sizes. It's just channeling all that movement to a smaller spot. Okay? So it doesn't take much vibration of the eardrum to cause a lot of vibration of that oval window. Okay. And so the purpose then of those middle ear bones is to amplify and concentrate the sound, because it's gone from a big area down to a little area. Okay. So do you see, if you are exposed to real loud sounds, that's massive. Lots of sound waves coming in, which is going to cause a lot of movement of the tympanic membrane, which causes massive movement of the oval window, which might set up too much fluid movement in the inner ear, and that can damage those little cilia that we'll see when we get to the inner ear. So you have a reflex that kicks in when you're experiencing loud sounds that helps prevent some of the movement of the bridge of bones. It's called the tympanic reflex, and it involves the contraction of two muscles. And one of the muscles is attached to one end of the bridge of bones, the other muscle is attached to the other end, and when they both contract, it pulls on the bridge and tightens it, which limits its movement. Okay. The two muscles have a name. This is anatomy. One's called the stapedius. It's attached to the stapes. That's easy. And then the other one at the other end, attached to the malleus, is the tensor tympani. So let's look at a picture of these. I've already talked about the eustachian tube. Okay, so here you see the tympanic membrane right here. Okay, so there's the malleus, there's the incus, there's the stapes. Okay, and here's the tensor tympani muscle right here. See how it's attached to the malleus? And then it's very difficult on this first picture to see the see the stapedius, but that's it right there. See how it's connected to the stapes? Here's a magnified view of it connected to the stapes. Okay. But those contract when this eardrum moves a whole lot, and that would be when you're experiencing loud sounds. Okay. And that could over time cause hearing loss. So even with the tympanic reflex kicking in, you all know you can have hearing loss if you're exposed to loud sounds for lengthy periods of time. Like if you work in an area where it's really loud, like at some factories, distribution centers are loud, they wear those ear protectors. At the airport, the guys that work outside wear the ear protectors because it's so loud. If you wear earbuds frequently, a lot of times that's too loud, and it can result in hearing loss. I have a couple of cousins who are audiologists over at Hudspeth, and they're seeing more and more hearing loss in children, and it's because of earbuds. It's a little scary. All right, that picture, this is a nice view, a sagittal section of the head, and of course there's a tongue. It's huge. There's the roof of your mouth. That's the palate. And then here's a nasal concha, and there's the upper one where the olfactory area is. This is the opening into the eustachian tube. Okay, that's highlighted in blue there. Okay, so it's up in that what we call the nasopharynx. Okay, I think this is a good time for us to take a break. So let's take about 15 minutes, and then we'll come back and finish up on the ear.